Serological detection of parasitic, fungal, and viral infections. So parasites are microorganisms that survive by living off other organisms, known as hosts. There's three main types: the protozoa, helminths, and ectoparasites. Protozoa are single-celled organisms that can live and multiply inside of human hosts. Helminths are going to be worms. They're multicellular, and their eggs are usually easily identifiable, as well as some of their body parts. And then ectoparasites would be multicellular parasites that live on the skin of the host. The immune response to parasites: innate defenses such as phagocytosis, cytokine production; adaptive immune responses, IgE antibody; possible outcomes: eradication of the parasite, death of the host, or establishment of a persistent infection. There's different ways that parasites can survive inside of our bodies. One of them is by antigenic concealment. That's when parasite antigens remain inside of host cells. So the parasite gets inside of your cells and very rarely escapes. And when it does escape, it's for such a short period of time that your immune system can't really detect it. Plasmodium, which is the genus that causes malaria. There's different species like Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae, and so forth. They cause malaria. They get inside of your cells, and they hide in there, and they can go undetected. Antigenic variation. That's when parasites vary their surface antigens. For example, trypanosomes, which alter their surface glycoproteins by gene switching, or leishmania, which have a complex life cycle forms. Antigenic shedding: it's when parasites shed surface antigens, which bind to the host antibodies and cells. Entamoeba histolytica is an example of one of those. So this guy is an intestinal parasite. It's a amoeba-type protozoan parasite, and it can actually eat holes through your intestinal wall. But it takes its outer surface, its outermost surface antigens, and when your immune system, when the antibodies bind to it, it just sheds them. So that immune complex lands in your tissues and causes a lot of local inflammation. Antigenic mimicry. Parasites express epitopes that are similar or identical to host molecules. Schistosoma can do this. So the surface proteins on schistosoma, and schistosoma causes schistosomiasis. It's a fluke, so it's a multicellular type worm that's a flatworm. And this guy has a very interesting life cycle that typically involves water and working its way through your skin when you're wading inside of snail-infested waters. But with this one, it gets into your bloodstream, or it gets into your liver or into your bladder, and it can actually put surface proteins on its back that mimic what your proteins look like. Immunological diversion. So parasites induce production of proteins that divert the attention of the immune system. Plasmodium falciparum infected red blood cells induce B cells to divide and differentiate into plasma cells. Immunological subversion. Parasites produce proteins that act as homologs of the immune system components. Complement cytokines, Fc portion of antibody, and HLA. Laboratory diagnosis of parasitic infections. Microscopic detection of the parasite in biological or tissue samples. So parasites physically can be found in various types of samples. In addition to that, there's a lot of molecular testing that occurs today. So with molecular tests, we look for the genetic material of the organism instead of just looking for the actual parasite. Because if the parasite is hiding somewhere in the body, then it's difficult to detect it. Serological methods would be what this class focuses on. So that's the detection of parasitic antigens or antibodies against those parasitic antigens. It's most useful when the detection of the parasitic antigen is difficult or not possible. So for example, toxoplasmosis. So toxoplasmosis is caused by an organism called Toxoplasma gondii, and this is transmitted through cat feces. And we'll learn a lot more of the various types of parasitic infections, but just to give you a little glimpse into this guy, usually not an issue. These things hatch open and they embed themselves in your muscle tissue, and you're kind of a dead-end host for the most part. But if a pregnant woman is exposed to this for the first time while she's pregnant, those little cysts can embed into the fetus and can actually cause death of the fetus. So that's when it becomes a major concern. Now, when this is inside of our body, we typically can't see it because those little cysts will embed in our muscle tissue. So we look for the presence of antibodies against it. And if we have antibodies, then chances are likely that we've been exposed to it. So toxoplasmosis, like I was mentioning, caused by the protozoan Toxoplasma gondii. There's 3 life cycle stages: the tachyzoite, bradyzoite, and sporozoite. Transmission to humans is through ingestion of raw or insufficiently cooked meat, ingestion of oocysts in cat feces, and ingestion can also be inhalation. So if it gets into your respiratory tract, usually that ends up just getting swallowed down into the intestinal tract. And transmission of tachyzoites across the placenta to the fetus. And this is the complicated life cycle of this. And you don't need to worry too much about life cycles in the immunology class. We will go in detail through these various life cycles once we get to talking about parasites and advanced microbiology. So incubation of 5 to 23 days. In healthy people, many people are asymptomatic. They may have mild lymphadenopathy or swollen lymph nodes. In immunocompromised patients, reactivation is a result of rupture of tissue cysts. Invasion of the central nervous system with encephalitis can be fatal. And then there's that congenital infection. Congenital means it's acquired through birth or while the baby is in utero. So that can lead to miscarriage, stillbirth, mental deficits. The risk is higher in the 3rd trimester. So if mom gets initially infected, or if the mom is infected and those cysts rupture and happen to spread to the baby, that's when the highest risk is in the 3rd trimester. Serological testing for toxoplasmosis determines whether the infection was recent or in the past. So if IgG is present and IgM is negative, that means it was a former or a previous infection. If IgG is negative and IgM is positive, it could indicate an acute infection or a false positive IgM result. So then you have to collect a second sample. If IgG is positive and IgM is positive, it indicates a possible recent infection or a false positive IgM. You want to perform an IgG avidity test to determine the cause. Fungal infections. So fungi are a heterogeneous group of eukaryotic organisms. They include yeast, unicellular forms that reproduce by budding, and mycelial forms, which is the multicellular form that reproduces through the production of spores or conidia. Mycoses, that's the scientific term for an infection, a fungal infection. So those are infections caused by fungi. Can cause hypersensitivity or type of allergic reaction or immune overreaction, mycotoxicosis, mycetismus, and tissue infections. Mycoses involving tissue infections can be superficial, cutaneous, subcutaneous, or systemic. Superficial are restricted to the outer layers of the skin. Cutaneous involve keratinized body areas. Subcutaneous involve deep ulcerated skin lesions in subcutaneous tissue. And systemic would be deep viscera and can spread throughout the body. Immune responses to fungi. Innate defenses include skin and mucous membranes. Recognition of PAMPs by PRRs, example, toll-like receptors. Cytokine production and inflammation. In the adaptive immune response, we have cell-mediated immunity. T cells and cytokine production is most important. And the role of antibodies in defense against fungal infections is uncertain. So laboratory diagnosis of fungal infections, we can isolate the organism from clinical samples. That's the gold standard, but it may be difficult due to slow growth. Some fungi take 4 to 6 weeks to grow, and sometimes we don't have 4 to 6 weeks to wait. Histological evidence of tissue invasion, skin testing, so we can do skin scrapings and we can look at the scrapings directly under the microscope. And then when we do that, we can dissolve away like human tissue using potassium hydroxide solution, and what's left would be the fungal elements. And then we can look at those fungal elements and say, does it look like hyphae? Is the hyphae septate, aseptate? And we can kind of try to go from there. Serological detection of antigens or antibodies, and molecular assays. Molecular assays detect fungal nucleic acid, but they're not yet widely available. Examples of fungal pathogens are listed here, and we'll go through each one of these. So Aspergillus, common species include Aspergillus fumigatus, flavus, and niger. They're recovered from soil and plant material, spores in indoor air, and they're commonly around in the environment. In immunocompetent hosts, they can cause external ear infections, pulmonary aspergilloma, or allergic bronchopulmonary aspergillosis. In immunocompromised hosts, they can be invasive pulmonary aspergillosis or disseminated aspergillosis. Laboratory diagnosis would be a positive tissue biopsy demonstrating Aspergillus hyphae, positive Aspergillus culture, or detection of Aspergillus antigens in the serum. Candida. Candida is a yeast that may exist as commensalistic organisms in human hosts. They're found on skin and in the GI tract and in the female genital tract. Candida albicans is the leading cause of infections related to Candida species. In immunocompetent hosts, it can cause diaper rash, vaginitis, and female UTI, and oral thrush. So we're talking about like vaginal yeast infections, oral thrush. And oral thrush is pretty common in babies with the yeast infection forming on the tongue. In immunocompromised hosts, oral thrush, esophagitis, pneumonia, and septicemia. Laboratory diagnosis, so we would try to culture it, the KOH prep of clinical material, and then there's serological assays. Look for mannan and anti-mannan antibodies. If you're colonized, it can stimulate antibody production in uninfected people. Immunocompromised patients may not produce detectable antibody. And then there's molecular assays. These are sensitive and specific for diagnosing early infections and invasive infections. Cryptococcus neoformans. This is an encapsulated yeast found in rotting wood and soil contaminated with bird guano or bird poop. Enters the host through the lungs, causing a range of manifestations from asymptomatic to life-threatening pneumonia. It can also disseminate to the central nervous system and cause meningitis or meningoencephalitis. The difference is meningitis, particularly around the spinal cord. Meningoencephalitis also involves the brain. The majority of symptomatic infections are in immunocompromised individuals. This is what Cryptococcus neoformans looks like under a microscope using an India ink prep. India ink is a negative staining technique where it stains the background, illuminating the organisms. And here you can see the organisms in the center, and they're surrounded by this halo, which is the capsule. Serological testing, use serum or CSF, detect cryptococcal polysaccharide antigen, antigen titers, latex agglutination, and LFA or lateral flow assay. And this is a picture of the lateral flow assay and what it looks like. Histoplasma capsulatum. It's a dimorphic fungus. Dimorphic means that it can grow in different forms at different temperatures. So at like body temperature, it would take on more of a yeast form, but in room temperature, it would take on more of a mycelium or more of a mold type form. And it's commonly found in soil in the Ohio River and Mississippi River valleys. It's acquired by inhalation of mycelial fragments and spores. And infections are mostly asymptomatic and self-limiting. Opportunistic infections are possible in immunocompromised people. Acute or chronic pulmonary symptoms are most common and can disseminate to organs rich in mononuclear phagocytes, and it's fatal if untreated. We can culture. Culture is dangerous because it is such a pathogen. But serological tests for H. capsulatum antibodies. So we do complement fixation and precipitation, which is the most common. The antibody to H glycoprotein in less than 10% of patients, but highly specific for active infection. the antibody to M glycoprotein in most patients, but cannot distinguish between current and past infection. Skin testing becomes positive 2 to 4 weeks after infection, remains positive for life. Detection of histoplasma polysaccharide antigen, urine tests high sensitivity and specificity. Coccidioides immitis. This is another dimorphic fungus that's prevalent in southwestern United States. Inhalation of Conidia may produce mild infection or symptoms similar to community-acquired pneumonia. Laboratory diagnosis would be serological testing, culture identification of the fungus in samples, immunodiffusion or EIA tests for IgM and IgG. So this, my aunt actually got. So she lived in Arizona and she loved to do a lot of gardening. This was commonly found out in Arizona. So she's doing her gardening and all of a sudden she got this major sinus infection that wouldn't go away. And it was very persistent and she tried antibiotic after antibiotic. She actually had a sinus biopsy and it was cultured and it still came back negative for bacteria and they didn't understand what all the inflammation was about. Once she told me all of these symptoms were happening, I told her, have them check you for different types of molds. Now the problem is she got the infection in Arizona right before she moved back to Wisconsin. Wisconsin doctors aren't really familiar with Coccidioides immitis. It's a pretty rare thing that you would encounter in people that live in Wisconsin. So this is where like patient history and patient information would have really come in handy. But sure enough, as soon as I told her have them check you for different types of fungal infections, she came back positive for this and they were able to treat her successfully with an antifungal. Her symptoms cleared up and she's doing much better today. And you can read through this summary.